Hello, we're glad you've joined us for this live webinar, Reducing Human Errors in Pharmaceutical Manufacturing Quality Control. I am Judy O'Rourke of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars, advancing scientific collaboration and learning. It's brought to you by Beckman Coulter Life Sciences. Beckman Coulter Life Sciences develops, manufactures, and markets products that simplify, automate, and innovate complex pharmaceutical quality control testing. More than 275,000 Beckman Coulter systems operate in both diagnostics and life sciences laboratories on 7 continents. For over 75 years, our products have been making a difference in people's lives by improving compliance and reducing cost of ownership. To learn more about Beckman Coulter Life Sciences, please visit www.beckman.com. Let's get started. You can pose questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them into the drop-down box located on the far left of your screen, labeled Ask a Question, and click on the Send button. Questions will be answered after the presentation. To enlarge the slide window, click on the arrows at the top right-hand corner of the presentation window. If you experience technical problems seeing or hearing the presentation, just click on the Support tab found at the top right of the presentation window. Or report your problem by typing it into the Answer a Question box located on the far left of your screen. This is an educational webinar and thus offers free continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process of obtaining your credits. I now present today's speaker, Tony Harrison, a leader in the pharmaceutical manufacturing quality control business for Beckman Coulter. Tony held the convenorship of the ISO working group revising ISO 14698-1 and 2 for microbial control in cleanrooms. He served as the UK subject matter expert to the ISO working group that issued the 2015 revised versions of the ISO 14644-1 and 2 documents for cleanroom classification at the heart of the aseptic manufacturing chapters of both the European GMP and the USA cGMP documents. Tony holds a bachelor's degree in electrical and electronic engineering and is employed by Beckman Coulter Life Sciences as a senior marketing manager. Experienced in water system total organic carbon, TOC, conductivity and ozone analysis and cleanroom environmental monitoring, as well as particle characterization, Tony has spent the last 15 years in applied metrology for the pharmaceutical and healthcare manufacturing industries. Prior to that, he worked for companies providing process control automation solutions for manufacturing industries. Tony was joint editor on the ISPE Guide to Ozone Sanitization of Pharmaceutical Water Systems and was also chief editor of the PHSS Best Practice Guide for Cleanroom Monitoring. Tony is a well-known international speaker and has provided educational seminars on TOC, liquid particle counting, ozone sanitization for water systems, and cleanroom monitoring worldwide. Tony Harrison will now begin his presentation.
Thank you very much for that introduction. My name is Tony Harrison from Beckman Coulter. Hello to everyone, and welcome to this short presentation on reducing human error in the pharmaceutical quality control department to help improve data integrity and also to help reduce costs. Today's presentation will be short, last approximately 20 minutes, plus, of course, the question and answers session at the end. This talk takes a look at 4 common quality control procedures and how automation can help improve data integrity by reducing opportunities for human error, whilst also saving time and reducing operator costs. The 4 quality control procedures discussed in this presentation form a common thread in the biopharmaceutical manufacturing industry and are water quality testing, clean room routine environmental monitoring testing, viable cell counting, and final injectable drug particulate testing. But first, let's take a look at the effect of human error in pharmaceutical quality control data integrity. A recent report published by CPHL highlights the pharmaceutical industry drive to improve productivity by improving right first-time quality, thus reducing manufacturing costs. Another report states that 79% of all 483 warning letters issued by the USA FDA in 2016 cited 21 CFR Part 11 data integrity issues. Whilst retraining is a common response to an FDA 483 warning letter, this may be only part of the solution. Was the problem simply that the technicians didn't follow the SOP correctly? Or was it really that the manual SOPs are simply not robust enough to prevent future recurrences of the same problem? Any process that automates the quality control SOP can potentially both reduce the impact of human error on data integrity issues and also bring welcome savings by reducing the time technicians need to devote to carrying out the SOP. Despite encouragement from the FDA in their 2004 Process Analytical Technologies initiative, many routine quality control test procedures in the pharmaceutical industry remain intensely manual and time-consuming. In the spirit of the FDA's PAT initiative, users could consider moving from sole reliance on laboratory-based QC testing to using the online QC instrumentation instead. For example, many companies have validated online water quality testing instrumentation on their purified water and water for injection loops, but they continue to rely on time-consuming laboratory water quality instrumentation for product release testing. As long as the online instruments can be validated and meet the requirements of the pharmacopoeias, it makes sense to at least reduce the amount of laboratory testing and to rely on data from the online instruments instead. As the online instruments are automated, their use makes the testing more robust by avoiding human error and thus improving data integrity and, at the same time, reducing QC department costs. A study by the Industrial Biodevelopment Laboratory showed more than 20% variability in test results between 16 experienced QC technicians performing the same manual standard operating procedure. Now, clearly the manual SOP could be improved and the technicians retrained, but such variability in results suggests that the manual process itself is just not robust enough to deliver the required level of quality control. Whilst the technical specifications of similar QC instrumentation may appear equivalent, when looking to improve quality control, QC teams should be looking at the level of automation in the instrument to make QC testing more robust and repeatable. For example, a common source of errors in QC testing is sample preparation. An instrument that automates the sample preparation process can bring enhanced reproducibility and robustness to a QC test. So let's take a look at our first QC test today: quality control testing for purified water and water for injection. Total organic carbon and conductivity are two of the four critical quality attributes defined for water for injection and purified water in the United States Pharmacopoeia. Online analyzers such as the PAT 700 from Beckman Coulter can be validated to comply fully for both of these key quality attributes according to the pharmacopoeial requirements. In their guidance, the FDA are keen to emphasize that the 21 CFR Part 11 ruling applies only to the data historian where the electronic records are kept. The danger with online instrumentation with their own local data historian built in is that they could attract the full requirements of the 21 CFR Part 11 ruling. Analyzers such as the PAT 700 avoid this issue by allowing the local data historian to be disabled, thus ensuring that it does not attract the full 21 CFR Part 11 requirement as a data archive for electronic records. Now, traditionally, data from online TOC analyzer is stored in a validated supervisory control and data acquisition, or SCADA system, or a distributed control system, making process quality improvements challenging and adding a further significant amount of change control requirements to these systems. More modern approaches keep quality critical data records in a separate, secure archive, leaving SCADA and DCS systems dedicated to process control only and more agile. In support of this, the PAT700 can be configured to automatically send PDF electronic records via secure file transfer protocol over Ethernet for review and batch release, thus satisfying the FDA's ALCOA requirements for electronic records to be accurate, legible, contemporaneously created, original, and attributable. Unlike laboratory water quality testing instrumentation, which utilize reagents and require manual configuration for each and every sample and frequent, often daily, calibration, the online PAT700 from Beckman Coulter requires no reagents. It automates the test protocol and only requires calibration once every 12 months. Calibration and system suitability testing standard operating procedures are automated, requiring no manual data entry, and pass-fail reports are automatically generated, eliminating manual calculations. Calibration standards automatically upload their certified value, lot number, and expiry date to the PAT700 using radio frequency identification tags. Electronic chips are located on the calibration bottles, and these are read by the PAT7 automatically using RFID. So, okay, let's move on to take a look at what can be done to automate routine cleanroom environmental monitoring programs. Now, of course, the GMP documents mandate the air quality conditions for biopharmaceutical production in cleanrooms, but in fact, the real danger is the microbes on the human body. Humans shed around 30,000 skin cells per hour, all of which are potential carriers of microbes. Unfortunately, we do not currently have technology to detect airborne microbes real time, so air particle counters are used as a surrogate. Discussions between myself and the environmental monitoring managers at facilities right across the world highlights an increasing trend where the burden of carrying out environmental monitoring is moving away from the QC microbiology team and over to the production staff for two reasons, I'm told. Firstly, microbiology staff are relatively expensive to employ to carry out such routine tasks. And secondly, it reduces the number of people inside the clean rooms, thus reducing the potential for product contamination. The downside, however, is that the production team do not have the same level of knowledge about routine environmental monitoring, and this is creating challenges in itself. In larger biopharmaceutical manufacturing plants, there can be teams of 10 to 15 technicians whose job it is every month to take thousands of routine environmental monitoring samples from the clean rooms. The standard operating procedures are manual, and at each sampling location they have to manually type the location name into the particle counter before they start sampling. Counters have to be manually configured following written paper standard operating procedures, and at the end of each day, the paper printouts from each sample location have to be photocopied because the printers in each particle counter are thermal printers, and the printouts fade over time. And then finally, the results from every location have to be manually transferred into electronic format one by one. In an effort to improve data integrity in routine environmental monitoring programs, Beckman Coulter have optimized their MET1 particle counting instrumentation specifically for pharmaceutical quality control use, building into the instrument design the capability for pre-configured electronic standard operating procedures, including the instrument setup and configuration, automatic pass-fail reports to GMP criteria, and generation of 21 CFR Part 11 electronic records straight from the instrument itself. The user simply selects the electronic SOP that's been pre-configured in the instrument and hits the Start button. The instrument configures itself correctly according to the SOP, It carries out the correct test and produces the electronic test record all automatically. This not only improves data integrity, but also can reduce costs. The technician no longer has to take time collating the paper records and manually transferring the data into electronic records. Additionally, by deskilling the use of air particle counters through automated SOPs, this process facilitates moving the responsibility for cleanroom routine environmental monitoring from qualified microbiologists over to the production staff, further reducing costs. So let's move on to take a look at how viable cell counting can be automated. In their 2003 guidance on the implementation of their 21 CFR Part 11 data integrity rule, the FDA used the acronym ALCOA, where they define good data integrity practice as creating records that are attributable to the technician carrying out the testing, are legible, are created contemporaneously, original, and accurate. Therapy products in the form of homogeneous cell populations are typically enumerated for concentration and viability based on cells' ability to exclude a supravital dye, such as trypan blue. Traditionally, this is done manually using a microscope and hemocytometer. But this method can be both time-consuming and error-prone. Originally designed for counting blood cells, the sample volume used in the hemocytometer method is typically just 100 nanoliters, and very small errors using this method caused during sample dilution, mixing, pipetting, and visual enumeration by the technician can lead to very large errors in results, as the final count result is multiplied up to report cell concentrations in viable counts per milliliter. One study revealed variances in the reported cell concentration results between each member of a team of 16 experienced and knowledgeable technicians using the hemocytometer method of up to plus or minus 20%. So there's the acronym from the FDA. ALCOA, attributable, legible, contemporaneously recorded, original, and accurate. The USP Chapter 1046 indicates that automated cell counting and viability instruments can provide a higher degree of precision and a more reproducible enumeration. These improvements are optimal if the cell preparation stage is automated as well, thus reducing the opportunity for technician errors. Additionally, if the automated cell counter can use electronic pre-programmed cell counting standard operating procedure libraries that match the manual SOPs for cell counting designed by the user, then results are more likely to be accurate and reproducible. As well as improving the precision and reproducibility, automated cell counters can reduce the cost of this quality control test by allowing the technicians to carry out other tasks while the counter automates the preparation and cell counting stages for each sample. Counters that can count from 96 well plates can save significant technician time, hence reducing QC costs. At the same time, by automating the cell counting SOPs, the method becomes more robust. Creating the electronic record contemporaneously improves data integrity, as the results are attributable, legible, contemporaneously created, original, and accurate as per the FDA's ALCOA guidance in their 21 CFR Part 11 document. Finally, we come to our fourth test here. Let's take a look at the requirements for particulate testing in that final injectable drug solution and how these tests can be made more robust. Now, although largely harmonized, the requirements for parenteral drug particulate testing do vary from country to country and also from product to product. The volume of the sample to be analyzed and the format the results are reported in varies from product to product. For example, the sampling requirements for small volume therapeutic protein products, such as vaccines, is very different to that of a large volume parenteral, such as an intravenous drip bag. Results must be calculated and expressed in the correct format. For example, counts per container for the small volume vaccine, or counts per mil, depending on whether it's a drip bag or not. Now, whilst general purpose liquid particle counting instrumentation can be used for the testing of particles in parenteral products, Beckman Coulter HIAC counters have been optimized for the application due to the wide range of complexity in the testing. The various compendial tests are built into the HIAC software, and the HIAC will automatically calculate a pass-fail result. As quality control teams tend to use their product brand names to describe the product sample under test, the HIAC particle counters allow the user to select the required test for each sample by selecting the product name from a drop-down window. The correct compendial test for each product is automatically configured on the HIAC, and there are no manual calculations. So, I'd like to summarize the key points of the presentation. Whilst the industry focuses on errors and omissions in 21 CFR Part 11 electronic records, the underlying issue is that the retraining may only be part of the solution. Manual SOPs are just not robust enough to prevent future recurrences of data errors and omissions. Quality control instrumentation that automates the quality control SOP can potentially both reduce the impact of human error on data integrity and also bring welcome cost savings by reducing the time technicians need to devote to carrying out the SOP. So, thank you for your time today. I'd like to take your questions now, and you should all have received an invitation on your screens to download my white paper on this topic. But I'm ready now to take your questions. Thank you.
Thank you, Tony, for your presentation. A quick reminder for our audience on how to submit questions. Simply type them into the drop-down box located on the far left of your window presentation window labeled Ask a Question, and click on the Send button. Tony Harrison will answer as many questions as time permits. Let's see, the first question is, how can the PAT 700 TOC analyzer from Beckman Coulter be validated to support its use for product release testing?
Yes, good question. In fact, in the ICH-Q2 document, the tripartite committee from the United States Pharmacopoeia, European Pharmacopoeia, and the Japanese Pharmacopoeia lay down the guidance on how an analytical method should be validated to demonstrate that it's suitable for its intended purpose. I have a paper on validating the PAT 700 TOC analyzer to ICH-Q2, and I'll send it out to you.
You mentioned electronic records in PDF format are automatically exported from the PAT 700 TOC analyzer. Surely PDF files can be changed, so how do you ensure that the electronic record is secure?
Yeah, it's a very good question, and in fact I'm challenged about that by a lot of people as I travel around the world. But in fact, PDF format is one of the formats recommended by the FDA themselves in their 21 CFR Part 11 compliance document. And their basic argument is that a PDF file can be viewed on Windows, on Android, on Apple, on so many different platforms. So the chances are that an electronic format in PDF, an electronic file in PDF format will still be able to be read in 10, 15 years' time, no matter what versions of Windows, Microsoft Windows we're on. So there's two main ways in which we achieve the integrity of this. Firstly, the PAT 700 uses a secure file transfer protocol, which is an industry standard protocol for secure data transfer. And secondly, the PAT 700 automatically applies a randomly generated password to the PDF file to prevent it being altered in any way.
Thank you. How does RFID technology prevent manual data entry and calculations on the PAT 700?
Well, firstly, the PAT 700 has all the standard operating procedures for calibration, system suitability testing, etc. They're all pre-programmed in electronic SOP format in the PAT 700. So when the user initiates one of these electronic SOPs, the PAT 700 automatically reads the calibration or the system suitability, certified values, lot number, and the expiry using RFID tags attached to each calibration standard bottle. As the SOP is electronic and automated, the PAT 700 automatically carries out the SOP and calculates a pass-fail result based on the criteria laid down in the pharmacopoeias. Now, this avoids both manual data entry and manual calculations.
I would like to once again thank Tony Harrison for his presentation. Do you have any final comments?
No, just to say thank you for everybody for their time today.
Well, thank you again. I'd also like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today will be addressed by the speaker via the contact information you provided at the time of registration. We would like to thank our sponsor, Beckman Coulter Life Sciences, for underwriting today's educational webcast. This webcast can be viewed on demand through October 2018. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.
Thank you. Goodbye.